So I chose to do cloning for my speech. Okay. So what is cloning? The term cloning describes a number of different processes that can be used to produce genetically identical copies of an identity. The copy material, which has the same genetic makeup as the original, is referred as a clone. Researchers have cloned a wide range of materials, including genes, cells, tissues, and even entire organisms such as sheep. Do clones ever occur naturally? Yes, in nature some plants and single-celled organisms such as bacteria produce genetically identical offspring through a process called asexual reproduction. In asexual reproduction, an individual is generated from a copy of a single cell from the parent organism. What are types of artificial cloning? There are 3 different types of artificial cloning: gene cloning, reproductive cloning, and therapeutic cloning. Gene cloning produces copies of genes or segments of DNA. Reproductive cloning produces copies of whole animals. Therapeutic cloning produces embryonic stem cells for experiments aimed at creating tissues to replace injured or diseased tissues. Okay. So do clone animals always look identical? No, they don't. Clones do not always look identical. Although clones share the same genetic material, the environment also plays a big role in how an organism turns out. Why are the potential drawbacks of cloning animals? Reproductive cloning is a very inefficient technique, and most cloned animals' embryos cannot develop into healthy individuals. For instance, Dolly the sheep was the only clone to be born live out of a total of 277 cloned embryos. This is very low efficiency combined with safety concerns presents a serious obstacle to the application of reproductive cloning. Researchers have observed some adverse health effects in sheep and other animals that have been cloned. These include an increase in birth size and a variety of defects in vital organs, such as liver, brain, and heart. Other consequences include premature aging and problems with the immune system. Another potential problem centers to the age of the cloned cell's chromosomes. As cells go through their normal rounds of division, the tips of the chromosomes shrink. Over time, they become so short that the cell can no longer divide, and the cell dies. This is part of the natural aging process that seems to happen in all cell types. As a consequence, clones created from a cell taken from an adult might have chromosomes that are already shorter than normal, which may condemn the cloned cells to a shorter lifespan. Indeed, Dolly was cloned from the cell of a 6-year-old sheep, had chromosomes that were shorter than those of other sheep her age. Dolly died when she was 6 years old, about half the average sheep's 12-year lifespan. Wait, sorry, go back. Sorry. And then, so there are many advantages to cloning, but such as the chance of curing certain diseases and being able to breed ideal stock for research and consumption. However, the disadvantages of cloning are seen by many too far outweigh any benefits that may be seen. Because of the risk-taking involved in cloning, it is a technology that many experts say may be better left alone, at least until it is better understood. I agree with that also. And then these are my sources. Thank you.